all of you. I know that you guys celebrated the New Year last month. So I hope you all had a great holiday season. Okay, so it looks like we have a few attendees here already. So without further ado, let's get started. Good morning, everyone. I'm Renjita Pillai. I'm Ren. Most of you are familiar with me through my technical emails. But a very warm welcome to all of you to BioVision's training session on assay kits. We are quite excited to provide this session for our partners and their technical support across Asia. So those of you are new to BioVision, we are a life science products company located in the beautiful San Francisco, California. We have around 13,000 products at this moment, and this number changes rapidly as we add around 100 or more new products every month. As you probably know, we have 5 different product portfolios: assay kits, ELISA kits, proteins, antibodies, and biochemicals. I'm very proud to say we are one of the leading manufacturers of assay kits, and all our assay kits are developed right here in our facility in San Francisco. As of now, we have around 1,000 plus assay kits, and our scientists are constantly working on new targets and expanding this portfolio further. Which brings us to the current training session on assay kits today and why we are doing this. So the reason behind today's training session on assay kits is that on a regular basis, we do get a lot of emails from our partners and technical support team regarding various aspects of the assay kit protocols, such as how to prepare samples, how to set up the assay, what kind of plates should I use, how to analyze the data, so on and so forth. You got the idea, right? So we realized there is a pattern, and these questions are being repeatedly asked. And we thought, why not we address it in a webinar and explain some of the basic aspects of the assay protocol so that you guys can manage these questions and answer them the next time the customer asks you about it. So based on these most frequently asked questions, the whole webinar is arranged as a question and answer session. We hope that this will help you to answer some of the most frequently asked questions from your customers. So without further ado, let me get started on the webinar. So today's agenda, I'll be talking about the assay kits, giving you an overview of what we have in this portfolio. I'll be talking about the different types of probes that we use in our assay kits, how to set up the assay, how do you prepare the samples, how do you store the samples, what volume of sample should you use in the assay, what kind of samples are compatible with the kit, what do you do if you have a high background, high sample background? Should I deprotonize my samples? So these are some of the common questions. So I'm going to discuss all of this today. Also, we'll be talking about data analysis, plate selection, instrument settings, as well as in the end, I will talk a little bit about the methodology or the principle behind the workings of our assay kits so that you can get yourself familiarized with those as well. So like mentioned before, BioVision offers a comprehensive array of bioassay kits, which can be broadly classified into target-based assay kits as well as physiology-based assay kits. So what are target-based assays? Target-based assays can be further classified into biochemical assays. So these are in vitro assays used to detect, quantify, or study the activity of a biological molecule or affinity of a ligand to a particular target. So these are the target-based biochemical assays. We also have physiology-based, cell-based assays. So when I say physiology-based assays, these are assays that are carried out in the cells under normal physiological state. So an example is cell-based assays, and we have a wide variety of cell-based assays as well. So these assays takes place within living cells that allow the detection of a cellular response to a particular drug or a particular analyte. So some of the examples of cell-based assays include cell proliferation assays, cell death assays, reporter gene assays, and so on and so forth. We have 3 different platforms available for detection. Most of our assays are 96 well plate-based assays. We also offer detection through flow cytometry as well as fluorescence microscopy. For today's presentation, we are going to focus more on target-based biochemical assays. So most of the presentation will be based on our assay kits from target-based biochemical assays. So as mentioned before, target-based biochemical assays are used to determine either the enzymatic activity or the concentration of an analyte in your sample, or to study the interaction of a ligand with a particular target. So that's what target-based biochemical assays are intended for. The methodology can be either enzymatic assays, it can be enzyme-coupled reactions, or it can also be based on chemical probes. So this I will explain at the end of the webinar, what an enzymatic assay or enzyme-coupled assay means, or what is a chemical probe-based detection is. Detection modality can be colorimetric or fluorimetric, as you have seen for most of our assays, or it can be both. Some of the assays can be both colorimetric or fluorimetric. And reading can be done either kinetic or in endpoint mode. So basically the difference between these two, kinetic and endpoint mode, is such that in kinetic mode, you would mix the sample or the standard with the enzymes, and you immediately start reading the reaction. Whereas in endpoint mode, you would mix the sample with the enzyme mix, and then you would incubate for some time, for the specified amount of time that is mentioned in the protocol, and then you would read in the endpoint mode. So it can be, some reaction can be is specified to be done only in kinetic mode, whereas others are specified to be done in an endpoint mode. So, like I mentioned before, detection capabilities or detection modalities of our target-based biochemical assays is determined by the type of probes that we use in these assays. So probe selection is very important for biochemical assays, and I think this information will help you understand a little bit more about our biochemical assays. As you can see, we use 3 major probes: WST probe, oxyred probe, or pico probe, and majority of our assays use either one of these 3 probes for the final colorimetric or fluorimetric detection. So WST-based assays are colorimetric assays. So if you see the probe that we use as WST, it's a colorimetric reaction with a maximum absorbance at 460 nanometers. And the very distinguishing feature of WST-based assays is that the final color development of the reaction is in the orange color range. So if it's WST, yes, the end product will be, or the final reaction color would be in the orange range. oxyred based assays can be, as you can see from here, it can be either colorimetric or fluorimetric. If we use an oxyred probe in our assay kits, the reaction can be read either in a colorimetric mode or fluorimetric mode. The colorimetric mode has a maximum absorbance at 570 nanometer, and this leads to a light pink to dark pink color development for the final end product. And the exact color depends on the concentration of analyte in your sample. We also have a lot of assays where picoprobe is used for the detection. And in general, picoprobe-based assays are all fluorimetric in nature with an excitation emission at 535 and 587 nanometer. For the picoprobe-based assays, the picoprobe itself is blue in color, and the final color development can be anywhere from blue to pink, depending on the concentration of the target in your sample. So if you see that we have used a picoprobe in our assay kit, so the final color reaction can be blue to pink in the samples. Then we also have other assay kits. These are reagent-based assay kits. You may be familiar with K410, the phosphate assay kit, where malachite green and ammonium molybdate, which forms a chromogenic complex with the phosphate ion, and it gives an intense absorption at 650 nanometers. So we do have other kits as well, which are reagent-based. Then there is K940, which is a potassium assay kit. So these are all reagent-based assay kits where the interaction of particular reagent specific for the target that needs to be analyzed is used for the final color development. So I'm hoping that this information is useful for you to identify our assays based on the probes used. Now let's take a look at how our assays are set up. So we have enzymatic activity assays, small molecule or analyte detection assays, or inhibitor screening assays. In general, most of our biochemical assays are 96 well plate assays and has a total reaction volume of 100 microliter. So this is the reaction volume in one well of a 96 well plate. So most of our assays, this is the format. So you would have 250 microliter of your sample, then you would make it up to 50 microliter with assay buffer, and then you would add 50 microliter of the reaction mix, keeping the volume, the final reaction volume to 100 microliter. So this is like a typical assay setup for most of our biochemical assays. Now let's take a closer look at how the actual assay is set up using a K589 a glycine assay as an example. So the principle of this assay is based on the oxidation of glycine through an enzymatic reaction to an intermediate, which is then detected by the probe, the oxyred probe, to generate an intense fluorescence, which is directly proportional to the amount of glycine in your sample. So this is a general principle behind this assay. So in this example, we are using human serum as a sample type, and it requires, or the protocol is recommending to deprotonize the serum before it is being used in the assay. So you would use a 10 kDa spin filter to deprotonize the serum samples. And you would filter the serum through the filter, and the flow through is what is used for the assay. The flow through or the deprotonized serum is then mixed with the reaction mix, which is then read on a spectrophotometer at an excitation emission of 535, 587 nanometer. So basically, this is how an enzymatic assay or a biochemical assay is set up. And this is this sort of a setup is very common for most of our plate-based assays. I'm hoping you guys can all still hear me very well. Yes? Okay, thank you. Thank you. Yeah, so let me continue with the presentation. So let's move on to sample preparation. So in general, most of our assay kits can be used to analyze cell samples, tissue samples, serum or plasma samples. So these are the most commonly used sample types. So I'm going to talk about these 4 types of samples in general. So coming to cells and tissues, and generally speaking, the protocol for tissue or cell homogenization goes like this. You can homogenize 1,000,000 cells or 10 milligram tissue in 100 microliter of assay buffer using a Downs homogenizer or mild sonication on ice. So you would have noticed that I have highlighted the use of Downs homogenizer or mild sonication on ice because a lot of the times we get this question from our customer asking, how should we homogenize the cell or tissue? Should we just incubate the cells or tissue in the assay buffer, or should we just use some sort of mechanical disruption to lyse the cells? And the answer is always you have to use some sort of mechanical disruption to lyse the cells. So you can either use a Downs homogenizer or you can use mild sonication. And I stress the point, mild sonication, you do not want to use any high amplitude, high frequency sonication. Mild sonication meaning low amplitude, short pulses. That's what you need. So if you have a Downs homogenizer, you can use that. It's a milder form of lysing the cells compared to sonication. So we would recommend a Downs homogenizer to sonication. But some form of mechanical disruption is always necessary to lyse the cells. Just incubating the cells in the assay buffer is not going to lyse them. So that's a very important aspect of cell and tissue sample preparation. Should you always use 1,000,000 cells or 10 milligram tissue? And the answer to that is we recommend using 1,000,000 cells or 10 milligram tissue because these are the numbers that gives us a consistent and reliable data. But if you think you can get good data with less number of cells or less amount of tissue, you can go ahead and try that as well. Just use these numbers as a guideline. You don't always have to use 1,000,000 cells or 10 milligram of tissue. You can optimize these numbers. Should you trypsinize the cells or not? That is another common question that we get from our customers. Sorry. And the answer to that is trypsin is not generally recommended, but there are situations when you have to trypsinize the cells. In that situation, it is fine. You can go ahead and trypsin. But the general recommendation for lifting the cells off the plate would be to scrape them off if you can. So if you can scrape them off, just manually scrape them using a cell scraper and lift them off the plate, put it in a tube, pellet them, and then add the assay buffer. But if trypsin has to be used in for some type of cell types, then yes, you can use it gently. There is no need to have single cell suspension. So you can just, as soon as they start lifting off, you can take it off the plate, pellet them in a tube, and then wash them twice in PBS to just get rid of any trace amount of trypsin from the cells. So when it comes to serum and plasma samples, sorry about jumping the slides. So when it comes to serum and plasma samples, we would recommend to avoid using anything that has EDTA or citrate. Preferably use heparin to collect plasma samples. Serum and plasma samples, they do not generally need any processing. It can be directly used for our biochemical assays. Some protocols recommend deprotonizing serum or plasma samples, and in that case, you can use a a 10 kDa spin filter or perchloric acid or TCA to deprotonize them. Okay, so why do we always recommend using assay buffer for homogenization? Why can't you use another cell lysis buffer? Are other buffers compatible with the kit? So this is another frequently asked question from our customers. Can we use RIPA buffer? Can we use cell lysis buffer? And the common answer to that question is no. You have to use the assay buffer that comes with the kit for sample preparation, homogenization, and lysis. And this is very important because the assay buffer has detergents, salts, and other reagents that are necessary for the reaction, and that makes the reaction work most effectively with maximum sensitivity and detection capability. So we recommend using the assay buffer that comes with the kit for sample preparation and homogenization. Also, other buffers such as RIPA buffer or other cell lysis buffer may have detergents such as SDS or sodium deoxycholate, which may interfere with our reaction. So we generally recommend to use only the assay buffer. Just as a side note, we always used to have Trypsinize 100 0.1% in our assay buffer as a detergent for lysing the cells, but in order to comply with European regulations, we are going to switch the detergent from Trypsinize 100 to Tergitol by the end of 2021. Fresh samples are always recommended, but we know that it is not always possible to use fresh samples. So in situations where you need to freeze the samples, for cells and tissues, I would recommend snap freezing them in liquid nitrogen as soon as they are off the plate and storing them at -80 degrees Celsius. If you have proceeded to homogenize and make the lysate from the cells and tissue, you can still store the lysate at -80 degrees Celsius. I would highly recommend to avoid freeze-thaw cycles. And if you're storing samples, do not store them for more than 4 weeks or a month. You may also add protease inhibitors, which are EDTA-free. We don't have any protease inhibitors added to the assay buffer. So if you want to keep the lysate for downstream applications, such as doing a Western blot or something like that, then yes, we recommend adding a protease inhibitor, which is EDTA-free. In the assay buffer before homogenizing the samples. Serum and plasma samples can be stored in aliquots at -80 degrees Celsius. Again, avoid freeze-thaw cycles, and storage is not recommended for more than 4 weeks or a month. Another commonly asked questions from our customers is, what is the exact sample volume that I should use in the assay? And so the answer to that is also very simple. We cannot recommend the exact sample volumes. And I think it is quite obvious why we cannot recommend an exact sample volume, but I will explain why. It is because the exact sample volume to be used depends on the concentration of the analyte in your sample. So from sample to sample, actually even the exact volume also depends on the person who is actually homogenizing the sample. Variation can happen from person to person as well, right? So depending on how well the samples are lysed or the cells and tissues are lysed, the amount of sample that is needed for the assay can vary. So what we can do is we can only suggest certain volumes, saying, try 1 to 50 microliters of sample in the assay. But we can never give you an exact sample volume to be used in the assay. So my recommendation would be to test different sample volumes to see what works best within the standard curve range and then go from there. So this leads to the next aspect, which is, what is a good preliminary test? When you get a kit, when you get a bioassay kit in your hand or in your lab, what would be the first, what would be the best thing to do? So the best thing to do is to do a preliminary test, to run the entire standard curve once and run a few samples at a few different dilutions. So, this way you're not setting up entire 100 reactions at the same time. You have to determine the sample volume that gives you the best OD or the RFU value that stays within the linear range of the standard curve. So we would recommend to run a preliminary test with the entire standard curve and a few samples run at few different volumes to make sure that the standard curve is working in linear and the sample ODs are within the linear range of the standard curve. And once you have this optimized, then proceed to set up the assay with as many samples as possible. And also the standard curve has to be run every single time you run the assay. So basically what I'm trying to say is you cannot use the standard curve from a previous time to analyze the sample later on. Will my sample work with the kit? So basically all of our kits, all of our biochemical assay kits are optimized for mammalian samples. So when I say mammalian samples, it will include humans, mice, rat, rabbit, pig, hamster, cell lines that are derived from mammals, etc. So the kit is primarily optimized for mammalian samples. And whatever samples that we have tested in-house, it is shown in our data sheet. So the data that is shown in the data sheet, it is being tested in our facility. If the sample is not on the data sheet, then it simply means that those samples were not tested when the kit was launched. Will this kit work on fish samples? Will it work on bacterial samples? And again, the answer to that is very simple. It could work on any sample type if the reaction catalyzed is similar in these species as well. So if we go back to the glycine assay kit example, glycine is the reaction that happens in the glycine assay kit is where glycine is oxidized to glyoxylate and peroxide. So if that is a similar type of reaction that is happening in fish or bacterial samples, then yes, the same kit can be used to analyze the amount of glycine in those sample types as well. So again, we have not tested the kits on any other sample other than what is shown on the data sheet. One of the most popular questions, I have to say this, is regarding sample background. So for most of our assays, we recommend running a sample background alongside the sample. This is mainly done to make sure that the signals that we see, the signals generated by the sample, is indeed generated by the analyte of interest and not from any other endogenous factors or a matrix effect or something like that. So we usually recommend running a sample background control along with the samples for most of our assays. But the commonly asked question is, why is my sample background as high as the sample? And what can I do to reduce the sample background? So let me try and answer both of these questions here. So one of the main reasons why the sample background can be very high is because of incomplete lysis. So let's say you are incubating the cells in assay buffer for 10 minutes and then just taking the supernatant and using it in the assay. You are not lysing the cells efficiently here. You are just incubating it. And like I mentioned before, efficient lysis will only happen if there is some sort of mechanical disruption happening. So either using a Downs homogenizer or a sonication. So an incomplete lysis can be one of the reasons why the sample background is high, because whatever OD value that you actually see in sample is also the same as the sample background. Another reason why sample background can be high is from sample matrix effect. So endogenous interferences can lead to the high sample background as well. In all these cases, how can you reduce the background? One is to make sure that the lysis is complete using a Downs homogenizer or mild sonication. Make sure that you are lysing the cells and using the supernatant after an efficient lysis. You can try and reduce a sample matrix effect by using a 10 kilodalton spin filters to deprotonize, or actual deprotonization using acids such as perchloric acid. Or you can also dilute the samples in the hope that the interference will be diluted as well. So these are some of the ways by which you can reduce the sample background. Sometimes there are rare occasions where the sample background will still be high, and in that case, I would just say that those types of samples are not compatible with the kit, and there's probably not much you can do with those type of samples if you have tried everything to reduce the background, and if it's still not working. Let's move on to deprotonization now. So this is another aspect of sample preparation that is most frequently asked. Like, why should we deprotonize the samples? Is there a reason? And the reason for deprotonization is that the presence of proteins in biological samples often interferes with the analysis of enzyme activity or analytes or small molecules. And so deprotonization is generally recommended for accurate determination of enzyme activity or the concentration of analytes in your sample. In general, BioVision suggests two methods for deprotonization. One is through the use of a 10 kD spin filter, or catalog number 1997, or deprotonization can be done using perchloric acid K808 or K823 TCA-KOH. And depending on the protocol, most of the protocols will specify what type of deprotonization should be used, because it has to be compatible with the protocol as well. So I would recommend to just read the protocol and decide which deprotonization method can be used. Obviously, 10 kilodalton filter is more easier, but it's more expensive as well. But 10 kilodalton filters are easier to do than PCA or TCA deprotonization. One thing I want to draw your attention is to the use of the 10 kilodalton filter. So if you're using a 10 kilodalton filter, depending on the molecular weight of the enzyme or the analyte that is detected in your assay, you should decide whether you should use the flow through or the retentate. Okay, so because you're using a 10 kilodalton filter, right? If the enzyme or the analyte has a molecular weight that is higher than 10 kilodalton, then you have to use the retentate for the assay. If the molecular weight is less than 10 kilodalton, it will flow through the membrane, and then in that case, you should use the flow through for the assay. So depending on the assay or the analyte that is being quantified, you have to determine whether you're using the flow through or the retentate for the assay. So we have a couple of recommendations when you deprotonize your samples using perchloric acid or TCA. The first recommendation, or the most important recommendation, would be to test your samples. Make sure that, so before you actually do the assay, just simply take a test sample and deprotonize it and see how that goes. So make sure that the suggested volumes, because if you use K808 or K823, the protocol comes with suggested volumes of TCA or PCA to be used with the sample. You have to make sure that these suggested volumes are indeed neutralizing your sample type. So do a preliminary test using a pH paper. So once you have neutralized your sample, dip a pH paper to see if the samples are actually neutralized, because you need the pH to be around 7.2 to 7.4, a neutral pH for the assay to work most effectively, because any leftover PCA can interfere with the assay. So this is a strong recommendation from our side. If you do deprotonization using the acid deprotonization using K808 or K823, then we would recommend doing a preliminary test to make sure the samples are indeed neutralized. Also, if the sample volume is not what is recommended in the protocol, then make sure that you can actually reduce the volume of PCA or TCA that is used. All you have to remember is to maintain the sample to PCA ratio and to use the suggested amount of KOH to neutralize. Also remember that deprotonized samples are diluted, so make sure you take that into account when you do the data analysis, that the samples will be further diluted if you are deprotonizing the samples. Generally, it is believed that using a 10 kDa molecular weight cutoff filter, it removes approximately 98% of the protein, and PCA precipitation at a PCA sample ratio of 1:4 removes approximately 95% of the protein present. Okay, so now have we reached the data analysis part of the presentation or the webinar? So now you have looked at sample preparation, sample storage, sample volumes to be used, deprotonization, how to set up the assay. Now you have run the assay and you have some data. How do you analyze this data, right? So let me go through the data analysis by taking a bunch of examples. I'm going to use K622 as a representative of one of the most commonly used quantitative assay kits we have. As you can see, all of our quantitative assay kits are provided with a standard curve for quantifying the target in your sample. So in here, the standards are run in an endpoint mode. And so at the end of the run, you take the OD value and you plot the standard curve. And if you're using an Excel, you will get a linear standard curve equation for the standards. So using this linear equation that is shown here, you would now have to calculate the concentration of the analyte. And in this case, it's a triglyceride. So the concentration of triglyceride in your sample. So basically, all you're doing is you have a standard curve, you're comparing your sample analyte concentration to that of the standard curve and calculating the concentration of sample analyte. So the equation, this is a generally commonly used equation or formula for calculating sample analyte concentration, is B over V times D nanomole per microliter, 1 nanomole per microliter or millimolar. So I think for most customers, the most confusing part is how to calculate this B in this equation. So B is the amount of triglyceride from the standard curve. I will explain how to calculate B a little later, but let me go ahead and explain what V is. V is the sample volume added into the reaction well. So this is not the total well volume. Remember, I was talking to you about the total well volume, which is 100 microliter. No, this is not the 100 microliter, but this is the amount of samples. So if you have only used 2 microliter of serum in the well, then the V is 2 microliter, not 100 microliter. D is the sample dilution factor. And again, it's very confusing for a lot of our customers. This is a dilution that is added to the V before adding it to the well. So let's say you have 2 microliter serum, undiluted serum, okay? You have not done any dilution and you added the 2 microliter of serum to the well. In this scenario, the dilution factor is 1. There is no dilution. But let's say the 2 microliter of the serum you have diluted previously before adding to the well, and the dilution was 1 in 10, then the dilution factor to be added in this formula will be 10. I hope you are I hope this explanation makes sense. If you have any questions, you can ask me at the end of the webinar. So basically, D is the dilution that is added to the sample before you add it to the well. It is not the dilution within the well. So it's not too diluted in 100 microliter. That is not the dilution. Okay. Okay, now let's see how you can determine B. So B is what is determined using the standard curve equation, which is in the format y equal to mx plus c. All you have to do here is solve for the x, which is the concentration. So x is equivalent to B in this formula. I know it's different letters, but x is what we are after. We are trying to find x in this linear equation. So you would substitute the OD value for y, whatever the protocol suggests, the OD value or the RFU value, or sometimes we also recommend to use the change in RFU or the delta RFU. So that would be substituted for y. M is the slope of the standard curve, and C is the y-intercept. And if you're using Excel, these values, M and Y, as you can see here, will be already generated by Excel. So in the equation here, all you have to substitute is the OD value for Y, and then you calculate for X. And whatever value that you get for X from this equation is equivalent to B. And you would substitute that value in this formula and determine the concentration of the analyte in your sample. I hope this is clear. Like I said before, if you have any questions, you can ask me later. Let's take a look at how this calculation would be if you have spiked samples. So the difference is if you are spiking your samples, then in that scenario, you are not using the standard curve. Basically, if you're spiking the sample, B is calculated in a very different way. Just take a look at this equation, and B is not calculated from the standard curve. B is just based on the difference between the spiked sample and the unspiked or the sample as such. So we are not, if you're spiking the samples, we are not using the standard curve to calculate B. Another example would be to calculate the enzyme activity. Here again, this is a quantification kit. So if you're calculating or quantifying the enzyme activity, the only difference here between this kit and the previous kit, the K622 and K227, in this kit, because you're quantifying the activity of the enzyme, you are also including the variable delta T, because enzyme activity is the amount of substrate cleaved by the enzyme in a minute or a particular amount of time. So the only difference between an enzyme activity kit compared to an analyte quantification kit is that in an enzyme activity kit, you're also incorporating the variable time to calculate the activity of the enzyme in your sample. Pretty much everything else is the same between enzyme activity kit and a analyte quantification kit. You are comparing the activity of enzyme to a standard, and you calculate the B based on the standard curve, B or the concentration of enzyme in your sample based on the standard curve. So these two types of kits have very similar data analysis. Then we have these inhibitor screening kits, which is a little bit different compared to a quantitative assay kits that we have. So in this third example, let's see how data analysis is done in an inhibitor screening kit. So here, this is not a quantitative kit. So there is no standard curve to actually compare your sample inhibitor activity. What you're doing is you're comparing you're not quantifying anything here. Basically, instead of quantification, what you're doing is you're comparing the activity of enzyme in the presence of the inhibitor to the activity of enzyme in the absence of the inhibitor. So basically what you're measuring is percentage relative activity or percentage relative inhibition compared to the enzyme control. You're always comparing the activity of the inhibitor to the enzyme. You can do this in two different ways. In the first method, you are basically just calculating the delta RFU of your enzyme control and your inhibitor, and calculating the percentage inhibition. In the second method, you can calculate the same percentage relative inhibition through calculating the slope of the enzyme and the inhibitor. So it's just like two different ways of calculating the percentage inhibition of your inhibitor. So that's all I have for data analysis. If you have any further questions, you're more than welcome to ask at the end. Let's move on to plate selection criteria for our assay kits. So generally speaking, for fluorimetric assays, you can use either a white plate or a black plate. It doesn't really matter. Each has its own purpose. White plates are generally used for amplifying fluorescent signals. So you would have seen in some of our assay kits, we recommend using white plates. And in some of our assay kits, we would recommend using black plates, but basically both are okay for fluorimetric assays, but do not use clear plates for fluorimetric assays. So clear plates are only used for colorimetric assays. And we recommend using flat bottom plates as opposed to U-bottom shapes for the assay kits. So for fluorimetric assay, white or black plate, both are okay. And for colorimetric assays, it's clear plates. And for both fluorimetric and colorimetric, you're recommended to use flat bottom plates. And the other thing is you don't really need any specialty plates like low binding tissue culture plates or some things like that, unless it is actually specified in the protocol. Sometimes we do provide with a UV plate. So unless it is not recommended in the protocol, you can just use a regular 96 well clear plates, white plates, or black plates. And I have listed some of the catalog numbers for these plates that you can use for the assays. Okay, so now that we know what kind of plates to use for our assays, let's take a look at the instrument settings needed for our assays. So as you know, most of our assays are 96 well plate plate-based assays, and we use, in our facility here, we use SpectroMax M2 spectrophotometer from Molecular Devices to measure the OD or the RFU. Softmax Pro is the software that is used for analysis and recording the data. If you are setting up a colorimetric assay, obviously the read type is going to be absorbance, and then you would select the wavelength, the appropriate recommended wavelength for the assay, for that particular assay. You can also select the 96 well plate that is going to be used for measurement. And for colorimetric assay, please always select a clear bottom, clear 96 well plate. You can, depending on what the protocol calls for, you can use an endpoint mode or a kinetic mode for measurement. And then you would, based on whether it's an endpoint mode or kinetic mode, you can choose the timing of measurement as well. Then at the end, you would select the wells to read, click OK, and this is how a final readout would look like for a colorimetric assay. For fluorimetry, it's pretty much the same, except the read type, instead of absorbance, you would select fluorescence or RFU. Then the next thing to be careful about is the excitation emission. I would recommend to keep the excitation emission as tight as possible. So let's say you have a 530 or like mentioned here, a 485, 570, a plus or minus 10 nanometer difference from the recommended wavelength is fine, but not more than that. Try and choose something as close to the recommended wavelength as possible. Generally, we don't use any cutoff filters, so that's generally left off. Nothing is selected for a cutoff filter. Usually the wavelengths are far apart that we don't really need, or we don't really worry about a cutoff filter. One other important aspect of fluorimetric assay is the gain setting or the sensitivity of the instrument. So usually for gain setting or sensitivity, different options are available depending on what instrument you use. So if you find that your RFU is very low, sometimes I get these questions from customers saying RFU is extremely low, what should I do? So in that case, the first recommendation would be just to see if the instrument gain setting is set at very low. Sometimes people don't realize when they set up the instrument, this sensitivity would be set to very low, so the RFU will turn out to be very low as well. So if it is set to low, you can increase this to medium or high, or even to auto. I would say keep it at auto, because then the instrument will gauge the signal strength and adjust the signal strength accordingly. So you can either put it at auto or low for most of your assays for gain setting or sensitivity. You can, with regards to auto mix, you can keep it on or off. It doesn't really matter. If you are keeping it on, then you don't really need to mix the wells or mix the reaction manually with a pipette. If you are mixing the reaction manually with a pipette, you need to be very careful in order not to create any bubbles, because bubbles can interfere with the assay. So if you are using a pipette, be extremely careful not to create any bubbles. You can turn the auto mix off if you are using a pipette to mix it. So depending on what you do, you can select these settings. So that's all about the instrument settings as well. Depending on the type of instrument that you're using, these settings will change. But basically, the basic idea is to make sure that the correct OD or the recommended OD or excitation emission is selected for the specific assay that is going to be measured. Limit of detection, this is another, I'm not going to go too much into the details of how to do it, because the slide itself is pretty self-explanatory. So we get a lot of questions as to what is the limit of detection for a particular kit. So I have used K375, the urea colorimetric assay kit, as an example to show you how we calculate the limit of detection. So basically, if you know the lowest concentration of standard and the maximum sample volume that can be used, which is available in our data sheet, you can calculate the lower limit of detection. Likewise, if you know the highest concentration of standard and the minimum sample volume, which is again, both of these parameters are available in our data sheet. If you know both of these, then you can calculate the theoretical higher limit of detection for that kit. So when you get a chance, you can take a look at this formula and see how we calculate the limit of detection theoretically for our assay kits. Again, another most frequently asked question is about the positive control. So for most of our kits, we try and provide a positive control. And the main goal of providing a positive control is to make sure that the kit is working. Okay, so the positive control is not provided to compare your samples. It is not a control for your samples. So therefore, we always request our customers not to compare the RFU of your sample to that of the positive control. because there are instances where the customers see the OD or the RFU value of the positive control going beyond the highest standard in the standard curve. And this is pretty common to see that. And so do not panic if you see it, because the reason for that is we don't keep the activity of the positive control same from a lot to lot. There may be minor variations in the activity from lot to lot. We keep the concentration the same. The amount of enzyme will be the same from lot to lot, but the activity may vary from lot to lot. So sometimes it is possible that you see the OD or the RFU of the positive control going above the higher standard. So what do we actually look for in a positive control? What we look for is to see whether the OD or the RFU value is going up with respect to time. So when you run the assay, is the positive control RFU or OD value going up with respect to time? That is the one thing that you have to keep in mind. If the answer to that question is yes, then yes, the kit is working. If the answer to that question is no, then there is a possibility that one of the kit components is not working. So it is very crucial that you run the positive control every time you run the assay. So this way, troubleshooting can be much easier. If we know that the positive control is going up in RFU over time, then we know that the kit is working. If it is not, then we have to come back and look at other variables as well as to why the kit is not working. So please do not compare the positive control to your samples. Please make sure that you run the positive control every time you run the assay. We also get a lot of questions with regards to the names of kit components, the concentration of kit components, the exact composition of buffers. Unfortunately, we are very sorry, but none of this information cannot be disclosed because it's proprietary to BioVision. But don't be disheartened. If you have a particular question where you want to know one of the kit components is going to interfere with the sample, then obviously you can ask us that particular question. So let's say if you want to know the presence or absence of a particular component in our buffer or in our kit, obviously you can ask us that and we will be able to let you know whether that particular component is present or absent in our kit. But if you ask us for, the concentration of the enzyme or the concentration of the positive control or concentration of buffer, then unfortunately we won't be able to disclose those information. Okay, we are at the tail end of the webinar. So what I'm going to do is I'm going to go through a few of our most popular kits and explain the principle behind the workings of these kits so that you can familiarize yourself with how the reaction happens and how we detect the analyte in your sample. So as explained in the very first or the second slide, the methodology can be based on either enzyme coupled reactions or enzymatic reactions. So let's take an example of K652, alanine colorimetric assay kit. Okay. So what you will see in our data sheet is this. In the alanine assay kit, alanine is converted to pyruvate, which is specifically detected leading to proportional color or fluorescence development. But what happens inside the well is what I have shown here. So this is the enzymatic reaction that is responsible for the color development. And this principle is based on an enzyme coupled reaction. So what is an enzyme coupled reaction? So an enzyme coupled reaction involves determination of an analyte or enzyme activity by coupling of one enzymatic reaction with another more easily detectable reaction. Okay, and the product of the first reaction, in this case, this is a pyruvate, it forms a substrate for the second set of reactions. So the first, there are two sets of reaction basically happening in this assay. So it's an intermediate formed, which is pyruvate, which then forms the substrate for the second set of reaction, leading to the generation of H2O2, which is detected by the probe, generating either a color or a fluorescence. So this is what an enzyme coupled, there are one or more reactions happening in the well. Or in simple words, there's more than one reaction leading to the generation of a color or a fluorescence at the end. So those are the enzyme coupled reactions. We also have reaction where the principle assay kits where the principle is based on a simple enzymatic reaction as seen in lactate colorimetric fluorimetric assay kit. Here lactate is simply converted to pyruvate and a peroxide, and the peroxide is then recognized by the probe, generating an intense color or fluorescence. And this color, color development or the signal, either the colorimetric or fluorimetric signal, is directly proportional to the amount of lactate in your sample. So basically, this is a very simple enzymatic one-step reaction, whereas this one is enzyme-coupled reaction. So that's the difference between the different types. We also have inhibitor screening kits. These are very simple again. Basically, in inhibitor screening kits, the methodology is based on looking at the activity of the enzyme in the presence or the absence of inhibitor. So if, for example, in K844 MMP9 inhibitor screening kit, you are looking at the cleavage of the substrate by MMP9 with the inhibitor and without the inhibitor. So without the inhibitor, it will lead to an increase in fluorescence when the MMP9 cleaves the substrate. But in the presence of the inhibitor, there will not be any increased fluorescence. The fluorescence, in fact, is quenched, or decreases. So basically, inhibitor screenings are much simpler. It's just a comparison of the enzyme in the presence and absence of the inhibitor. Also, remember I was talking to you about chemical probe-based assay in the first or the second slide. I was talking to you about target-based biochemical assays, where the methodology or the principle is based on chemical probes. And one such example is the zinc colorimetric assay, K387, where the presence of zinc ions in your sample is detected by this specific ligand that is specific to zinc ions. So the ligand and the zinc ions are bound to the ligand, generating an intense color at 560 nanometer, leading to the detection of zinc in the samples. So these are chemical probe-based assay kits that we have. So overall, like I mentioned, we have enzyme couple assay kits, enzymatic reaction single-step assay kits, inhibitor screening kits, chemical probe-based. So these are the different types of assay kits that we have on target-based biochemical assays. So I know that you can always contact us for any technical questions, but we have a few pointers as to what or how you can contact us. Before contacting BioVision technical support team, please try and read our data sheet to see if the answers are available in the data sheet. You can check our FAQs on the product page. Every product, most products, we are trying to add more FAQs on a regular basis. So if you can, then please check our FAQs on the product page. If you have decided to contact us, I would say, please get more details of the problem. For example, just telling us OD values not increasing is not very useful information. If you can get more information about the problem, then it would be more beneficial for us in troubleshooting. And if you can include those details in your email to us, that would be great as well. When you contact BioVision, please, these are some of the the factors that we look forward to have. Please share the lot number. Please share analyzed data with the standard curve drawn. Please do not send raw data of standards or samples. If we don't know what we are looking at, it is very difficult for us to troubleshoot. So if you're sending data, please send analyzed data, averages and not raw data. If you can include information, so this goes back to getting more details from the customers. If you can include more information on the sample type, homogenization process, how old the samples are, how the samples were stored, is a positive control working? So include positive control data as well. So this will give us a indication that either the kit is working or not. So so this way the troubleshooting becomes more efficient. We can solve a question in a couple of emails rather than many emails. So these are some of the pointers that we would like to give to the technical support team. This is our contact information. This is the team that does the technical support from BioVision. I think you probably have listened to webinar from most of them. So I think the names are familiar to you. We are always here to help you. So anytime you have questions with regards to any of our product line, please don't hesitate to contact us. Last but not the least, thank you for your business and thank you for your wonderful partnership. We look forward to working with you again in the new year. Also, there's a small announcement. The next webinar will be on March 25th on BioVision's Molecular Biology Tools by Dr. Maithili Angela. So please keep an eye on for the email with regards to that. Thank you so much for listening to this presentation and for participating. If you have any questions, you can start asking. If you want me to go back to any of the slides, please let me know. I can go back to the slides as well. Presentation, the link to the webinar will be sent to you very soon. Any questions at all? Can you still hear me? Yes, yeah, we will be providing the PowerPoint very soon. So our marketing team will be sending out the PowerPoint very soon to all the distributors. And please forward it to your technical support team as well so that they can take a look at it and refer to the presentation whenever needed. Please don't hesitate to ask us any questions in future as well. These are just pointers. The presentation, the main idea behind the presentation is such that when you get questions, you can try and troubleshoot, and that way you can minimize the time, the turnaround time, if you already know the answer. So that was one of the goals, to increase customer satisfaction and also to decrease the turnaround time so that you can answer them faster. So yes, the presentation will be sent to you very soon. Any other question? If there is any other question, you can type it in the chat box, in the chat window, and I can answer them. So did you mean just the assay kits or in general, other products as well? So we do have some, we don't have any videos, but we do have some flyers for assay kits. I'm going to give you the email address. You can always email marketing@biovision.com for flyers and emails regarding flyers and brochures, actually, flyers and brochures regarding a particular product line. You can email marketing@biovision.com. Any more questions at all? If there are no more questions, then I would like to thank you all for participating in this webinar. For all additional questions, as always, you can email tech@biovision.com. And thank you so much for coming to the webinar. Bye, everyone.